All right, genetics, study of hereditary things to come, right? Genome is, like I said, it contains 23 chromosomes, 22 or 26, 23 pairs of chromosomes. 22 are somatic or non-sex chromosomes, whereas one pair is a sex chromosome pair. Now, we could use, as you're learning, I'm always saying this, to say one structure, you gotta know how to say it about 12 different ways. So an autosome is a somatic chromosome. It is also the non-sex chromosome. They all mean the same thing, okay? So these are the 22 pairs of non-sex chromosomes. We're using the words autosome or autosome. We also have the sex chromosomes, and females have what we refer to as 2 X chromosomes. Males only have 1 X, but they do have something called a Y. And that's what makes a woman and a man. A man is the XY versus the XX. Okay? Bruce Jenner, no matter what he says, he's an XY. Okay? In other words, he's going to get certain diseases that only XY people get. Okay? Women have a greater chance of having breast cancer. He would, well, although taking all the estrogen and stuff. But do you see what I'm saying? X, that's what it is to everybody. You go by this. Okay? And then whatever he wants to do with his life, that's fine. But he's still considered an XY in the science department. Okay? So this is what we call a karyotype. We want to figure out if there's extra, we can check to see if there's trisomy that we mentioned before, if it's female or not. They will put this all together by taking the chromosomes, by getting cheek cells, and they can go in their microscope, and they can go and figure out how they separate them. But this would be a female, this would be a male. They could actually look to see if there's an X chromosome here. The Y is usually shorter, and that's how they would be able to tell. Whereas here they have 2 X ones. How they go about doing this, each one is weighted differently, and it goes through certain machines that they could actually filter out which ones are the heavier ones and so forth. But those are what we call karyotypes. It's just the chromosome analysis of that particular person. All right, now, before we get into genetics, some of you had genetics and some of you haven't even touched it at all. So we got to just do some basic terms so we know what we're all working on the same page. There's something called an allele. An allele is 1 or 2 versions of a gene, and it can be more than one version. A gene is eye color. An allele is blue eye color, green eye color, red eye color, brown eye color. So we have 4 different alleles that I just mentioned to the gene of eye color. Does that make sense? Okay? You inherit 2 alleles, one from mom and one from dad. You got an allele from dad who has brown eyes, and you received an allele from mom who has blue eyes. You can see where this is going. What's the prediction of me getting brown eyes or blue eyes? That's what we're going to talk about. Homozygous is when the 2 alleles are the same for the gene. I got brown eye color from dad, his gene, but I got brown eye color from mom, from hers. Both of them are the same alleles. We say it's homozygous. Heterozygous is when they're different. Got mom's eye color gene, or allele rather, and it was blue, and I got dad's that was brown. They're different. Then we say it's heterozygous. Okay? A genotype. A genotype is what the actual inherited set of genes, the instructions. And you can't see that. It's what is in the chromosome itself. And I'll show you by giving examples as we go along. The phenotype is what actually is observable. So the genotype is getting, let's say, blue eye color from mom and brown eye color from dad, but my phenotype is I got brown eyes. It's how it's going to present. Autosomal means that we're talking about a gene that's on one of the 22 pairs of non-sex chromosomes. And sex-linked is some gene that's located on the X or the Y chromosome only. Now, we've got to talk about dominant versus recessive. And a dominant allele is an allele that you got from mom. Let's say brown eye color or blue eye color. And I'm just giving you, because I'm not too sure which one's more dominating offhand. But let's say the brown is more dominating. So if you got brown eye color from dad, we say that that's the dominating allele. In this particular case, the brown is. The blue is not. It's a weakened one. Okay? We represent this with arbitrary letters. We could say blue eye color and use B, or we could use E, we could use H. The letters have no relevance whatsoever. I personally like to use letters for teaching purposes and for myself, where the capital letter and lowercase letter don't look the same. Like H and H, they don't look the same. But if I put S and S, right? If I put an S here, is that lowercase or uppercase? See what I'm saying? All right? Uppercase or lowercase? All right? So teaching purposes, I'd rather use that for that reasoning. Does that make sense? Okay? All right. So a dominant allele is the one that's going to actually have enough force to show up on the person's face or on the person's lifetime. Okay? We could actually have it where we could have a capital H from mom and a capital H from dad. And let's say capital H is brown eye color, lowercase h is blue eye color. Lowercase h is what we call recessive. It's the weakened allele. If you got brown eye color, capital H from dad, and brown eye color, capital H from mom, then you're going to have brown eye color. If you got a capital H brown eye color from dad and a lowercase eye color of blue from mom, the H is the more dominating one, the capital H. So the brown eye color is going to show up, but you carry the trait for the blue eye color. The only way you can get blue eye color is that you got to get the lowercase h from mom and lowercase h from dad. Now you'll get the blue eye color. Does that make sense? Okay? So the trait is only expressed if you get that recessive gene from both mom and dad. Who here has done Punnett squares? This is going to be a breeze. But I'm going to do it as though you haven't had it, okay? Punnett squares are going to help you predict what that person's going to have, like the blue eye color or brown eye color. Okay? And we use letters, like I said, but arbitrarily. Now, autosomal recessive traits, what we're more concerned about is the diseases. We don't care about brown or blue eye color unless you're really aesthetic and you want your child to have that. All right? But you really want a healthy child. A good example of an autosomal recessive disease is one that we talked about, cystic fibrosis. Also, sickle cell disease is another one too. There's a whole slew of them. So what happens here is that if it's and I'm using C. I know that's breaking my rule about C, capital C, lowercase c, you can't tell the difference. But I'm going to have a picture over here, and the picture came from your book, and it uses C's. However, they're using the color, so you can actually see a difference. Okay? So a capital C, which is in green, and capital C that's green over here, that's saying that you're homozygous dominant. You don't carry the disease whatsoever. You're not getting the disease. You're not a carrier at all. completely normal. Then you have a capital C and lowercase c. See it's red? All right. This one I kind of accepted because they have different colors. But in this case, he's going to appear normal, but he's carrying the gene for it, the trait for it. And the only way you're going to have the disease if you have lowercase or red from both mom and dad. Okay? So what happens here is this. We do this little, what we call Punnett square. And if you've done Punnett squares before, we're only doing 2x2s. You know what I'm talking about, all right? We're not going into this 4x4s, and it's, all right? We're keeping it very simple, okay? This is mom. That's the abbreviation for female, okay? This is the abbreviation for male. Let's say mom is a carrier. So she's going to have a capital C from her mother and a small c from her father. Likewise, dad is also a carrier, and he had a capital C from his dad and a lowercase c from his mom. So what's the chances of the baby actually going to get the disease or be a carrier or completely normal if we know the genotype of mom and dad? The phenotype is that they look normal. You can't tell if they're a carrier or not, okay? But the genotype is what I put in the C's here. So you just combine things, you crisscross things. That'll be a CC. This will be a C, lower c. This will be a C, capital C, lowercase c. And then this one will be lowercase c, lowercase c. Does everybody understand how I did that? Yes? All right, crisscrossing them. And that's what's over here. So in this case, the chances, if mom and dad are both carriers, there's a 25% chance that the child will be completely normal. There'll be a 50% chance that the baby will be a carrier just like mom and dad. Not too much to be concerned about. But there's a 25% chance that the baby's going to have the full-blown disease. So if mom, and like I showed you before, 1 in 20 Caucasians carry this gene. So we check this in every pregnant woman. Well, actually we do it basically on everybody now. Jewish, blacks, everybody, because there's so much interracial marriages and stuff. Okay, so we don't know. Same thing with sickle cell disease, okay? So what's going to happen here is if mom, we test mom, she's pregnant, and we lo and behold, she has the gene for it. She's a carrier. What's the next test you're going to do? Check the dad, okay? If dad has it, there's a 25% chance that the baby's going to have it. Now this is where you have to, as a nurse or a medical professional, you have to step out of the light, okay? You can't tell her what to do and what not to do. This is where 25% chance the baby could have sickle cell disease or cystic fibrosis, whatever we're talking about. So if she chooses to want to abort, she doesn't, maybe she's had relatives who had sickle cell or had relatives with cystic fibrosis and she doesn't want to, or both of them don't want to have their child go through that, then that's their reason if they want to abort. We could do an amniocentesis, which is taking some of the fluid around the baby with a needle and syringe, and the baby's cells come off their skin, like dandruff, comes off their skin and floats in that fluid. So you're going to take some of that fluid and there's skin cells in there, and we could do a karyotype, and they could actually figure out if the baby actually has cystic fibrosis. But there's a chance for that too, that there could be something, you know, you do the amniocentesis, and you can have premature delivery. Rare, but it can happen. But it's up to the mom and dad to decide what to do. You can't say, oh, I wouldn't do the abortion if I were you. You'd always give it up. You can't do that. It's called patient autonomy. So if she has the trait and he has the trait, I'll give a little spiel about what cystic fibrosis is, but then I lead them to genetic counseling and let them talk about all the pros and cons of all this. And if they decide to do amniocentesis, then I'll talk to them about the amniocentesis and the disadvantages and advantages, and we'll go from there. But you can't put your own morals with someone else's, not, at least not in America. Okay? Now, with that said, if we do this again, and dad, I'll do it very quickly over here, and dad is not a carrier, so we're saying that mom, mom is a carrier, but dad is not. He's completely normal. So we find out that she's a carrier and he's not. There's no reason to do anything else, because look what happens over here. Now, the child's going to appear normal no matter what. 50% it'll be just like mom and she's doing well, and 50% chance that the baby's going to be completely normal. So there's no reason to do amniocentesis or consider abortion or anything like that. Okay? You understand how they would use this? Okay? Alright, so that's what's shown on here. Now, with that said, predicting the offspring, as I just showed you, so we're using this one up here. Alright? Genotypically, you will have 25% completely normal. Genotypically. We'll have 50% that will be carriers. And 25% will have the disease. Phenotypically, different story. 75% will appear normal because of the dominating allele. And 25% will have the disease. Remember, phenotype is how the person's going to look. What's the observational trait that you're going to see? Okay? Sickle cell disease, just to give you an idea, red blood cells are shaped like this. We'll talk about those later. Carries oxygen from one point of your body to the other point of the body. They should be round like this. In sickle cell disease, they become these crescents, these croissants, these bananas. Sickle, if you know what the old Russian flag looked like. Soviet Union had that sickle on there. Sickle, that's where they come up with that. Cut the grass. Okay? When they go into this shape, oxygen has problem binding to this area. So now these people aren't able to, they start clotting in the blood also. And you can't get oxygen from one point to the other point. And they go through these sickle cell crises and stuff. If you have sickle cell disease, I would say, my guess is about, and don't quote me on this, but maybe 60, 70, 80% of your cells turn into that. And they gotta go through blood transfusions. The life expectancy, they're getting better with it, but it's still not good, like 30, 40 years old, okay? It's just the body, it takes its toll. It's pretty tough. Sickle cell traits, they appear normal, but you do have sickle cells in the blood. Not that much, maybe 10%. And you could live to a ripe age of 120 if you can, with 10% being that bad, okay? Beta thalassemia is the hemoglobin itself. The chains, there's 4 chains in a hemoglobin. One of them can't be made, and it's not binding to oxygen in a certain way. What sickle cell is that there's one of the chains, it has, I think, about 150 amino acids in there. So you have 150 times 4, so you're looking at roughly what? Nearly 600 amino acids in one hemoglobin molecule. One amino acid, and I forgot which one it is, but valine is replaced by something else. That one difference makes them look like this. Okay? And thalassemia, you're missing the chain. So another one gets replaced, but it's not the proper one. All right? There's different types of hemoglobin. Fetal hemoglobin, adult hemoglobin. That's what happens. It has to do with hemoglobin. Okay? And there's a thalassemia minor and thalassemia major. As much as we have sickle cell disease and sickle cell trait, there's a minor one. Okay? So this is what red blood cells would look like, and their hemoglobin looks here. There's the four chains I was telling you about. There should be 2 beta chains and 2 alpha chains. But in thalassemia, they would have 3 beta chains instead of 2 and 2. All right. Here in sickle cell, their cells look like this, and their hemoglobin is kind of squished, and it's difficult to get the oxygen on there. Okay. So I did something similar to up here. Okay? So if you do a pedigree on this, now this is what a pedigree is all about. We can do this going backwards. Females are circle, that's what we usually use for pedigrees. Females are circles, and males are square. Square, okay? If it's colored or shaded, they have the disease. If it's not shaded, then they don't have the disease. If it's half and half, they're a carrier. Okay? That's the nomenclature of this. So when you look at this, which child is homozygous dominant male? John or Justin? Give it a moment. Don't call it out. Think about it. Now that you understand Punnett squares, and now the nomenclature of pedigrees, you could go backwards and figure out. Which child male is homozygous dominant? Who says John? Who says Justin? Yeah, good. Justin. Not too bad. Okay? Because the dominating one is not the diseased one, and it's clear up there. All right? So far, so good? All right, let me do this last thing here, and I'll give you guys a break. Sex-linked traits, diseases. If you understand this, then we're just going to kick it up a notch. All right? There's something else we just got to add to this, and it'll make sense. All right, sex-linked traits or diseases. Some genes are just on an X or just on the Y. All right? We were talking about those 22 pairs before. Now there's some things that are on the X or just on the Y. If this disease is sex-linked, it almost always involves the X chromosome. So therefore, we sometimes refer sex-linked and we call it X-linked. The reason why it would only be on the X-linked, we don't have, let's put it this way, we don't have vital genes on the Y chromosome. Stuff to make you breathe and heart rate and stuff. Do you know why? Because half the population doesn't have a Y chromosome. Does that make sense? So if they don't have a Y chromosome and there was something about heart beating on there, they wouldn't be surviving. Okay? Simple as that. Okay? The things that are on a Y chromosome is really pertaining to a male. Let's say testicular cancers or testicular diseases, infertility, something that's really pertaining more to males. Does that make sense? Okay? So the Y chromosome contains few genes vital for life because half the population doesn't have it. Okay? So most defects on the Y chromosome, like I said, you have testicular development, male infertility of that nature. So males only have one X chromosome, which makes this interesting. This is where you ladies are better than us. You start getting less diseases than males. You see, if women have two X chromosomes, if one of them is bad, has a bad gene on it, the other one is going to dominate that other one. You guys are lucky. Kris Jenner's not. Do you see what I'm saying? Okay. Males, if there's something wrong on their X gene chromosome, or there's a wrong gene or a disrupted gene on the X chromosome, they don't have another one that'll dominate it. So they would get the disease. Women could be carriers, but exceptionally rare that they would get the disease. Okay? So only one defected allele will affect a male. Okay? Alright. A good example of this is color blindness or hemophilia. Hemophilia is that when you get into AMP2, you learn about clotting factors to help you clot blood, and hemophilia is that you're missing one of those clotting factors. They actually did a pedigree of the English princes and the royalty that's going on over there, and they actually led back to one of them actually had hemophilia when he did this pedigree and stuff. But anyway, colorblind is a good example too. So this is where it gets a little bit more confusing. Not too bad, okay? So, let me draw you this one. Again, it's just a 2x2, same thing. Here's mom, and here's dad. Okay? So he's the same colors as up here. Okay. Mom is a carrier. So she has an X that's good. And there's also going to be an X that's recessive, that's the disease. Okay? Dad will be completely normal. He's going to have an X and he's no problem. And he'll also have the Y that will have, well, there's no R there really. We're just dealing with the X.
I think flipped it.
What's that? You doing it?
You flip the on the chart, the Y's on top. You have the Y on the bottom. That's all.
Oh, okay, all right. I see what you're saying. So, I mean this thing, right? So this is Y here. It doesn't matter when you do this, but that's I'm going to try and match that though. Okay. So here you have mom, and she looks completely normal because this one's going to dominate that. Capital R, lowercase r. Okay? Dad is completely normal too. He's got his X that's fine, and the Y is, that's not where the gene is. But look when we put this together. We have this, right? Crossing these over, and this will cross over. Then you'll have this, this, and take a moment, just let it register, and I'll talk about it. See, I did the crossing over and stuff. So what happens here is this. Dad is not a carrier. He doesn't have the disease. Well, he could never be a carrier. He either has the disease or not. Okay? Mom is a carrier. They mate. I guess that's the proper word. Okay? They mate. There's a 50% chance that you're going to have a boy or a girl. XY, XY, XX, XX. Right? That makes sense. 50%, it'll always be 50%. Alright? Even in Bruce Jenner's eyes, it will always be 50%. Can't change that. So what's going to happen here, though, is that you're going to have a completely prediction. A completely 25% of your offspring will be completely. Well, no, I'm sorry. 50% of the males that you would have is normal. Does that make sense? 50% of your females will be completely normal. XX. 50% of your males will have the disease. Females of your daughters will be a carrier. Okay? So in a case like this, there's a 25% chance that your offspring will have the full-blown disease. Does that make sense? It's confusing here. So that's this first generation, all right? So what's written here, completely normal male is this. Diseased female is over here, where it's carrying the X. Oh, sorry, diseased male, XY. This is completely normal female. And then this would be a carrier female. Just what I had up there. Okay? But let's do another generation. Mom's a carrier. The disease will appear in this generation. Does that make sense? Now, let's do another generation. Mom, in this case, and dad over here, mom is completely normal, is not a carrier at this pace. Okay? But this boy grows up and he mates with her. Oh, mate's the right word. And is going to be that, right? Now we put it together. Look what happened here. 50% of offspring, completely normal. 50% of the offspring will be carriers. But now look at the gender. 100% of the males, the sons, will be completely normal. 100% of the females, the daughters, are going to be carriers. Does anybody in this generation have the disease? It skips a generation. Let me give you an example, which is the truth. My father's colorblind. Okay? He's colorblind. What's my genotype? Am I a carrier? Am I colorblind? I'm completely normal. Well, I just went to the family reunion. Believe me, I went to the family reunion. I'm normal. What about my sister? Does she have the disease, a carrier? Is she normal? What?
Carrier, right?
100%. I can tell you right now, she's 100% she's a carrier. So it skipped my generation, but it went to the next one. And my nephew and niece, from her, it seems like they're fine. So they were in this situation here. See? So there's a few different caveats that actually happen with this X-link or sex-link that are pretty interesting. Yeah?
But it almost skips a generation if the fathers.
Correct. Yeah. What she's saying is if you carry the gene over here. Right. It's rare though, when you deal with these diseases, you'll know if you're a carrier and stuff. Yeah, it's rare. This is extremely rare that a female would actually have the disease. Very rare. Multiple muscular dystrophy is an X-linked gene. And when I mean growing up, I actually saw like the Labor Day telethon. That's the one that's coming up soon, right? Growing up, it's Jerry Lewis's, I don't know if you know who Jerry Lewis is nowadays, but I did. But Jerry Lewis, and he started, it's Jerry's Kids. I don't know if you ever see that, go to 7-Eleven, you want to put money in the donations, stuff like that. Now it's not donations, now it's tips at 7-Eleven. Where did that come from? Anyway, so what that was originally was for muscular dystrophy, MD. And growing up, I never really thought about it, but sometimes, a lot of times during the telethon, they would have some of the patients or some of the people with MD to come up. And now I'm looking back on it, it was always boys that went up there. It was never girls. And now I understand why. However, now I'm looking at the telethon nowadays, like you see it on Labor Day, and when they put people up there, we're seeing a lot of girls, women, men. It's not like autism is getting, there's a rampant, we're seeing a lot of bigger risk of it. You can't fight the genetics. It doesn't work that way. Then I found out what MD is, or what this telethon is doing. It's all muscular diseases that they're putting in there now. It's not just MD anymore. It's Lou Gehrig's disease and things like that that are more prominent in both. So that kind of threw me for a loop at first. I was like, I'm telling the students this thing, and I'm seeing them up there, and I looked it up, and it's a lot of muscular diseases they throw in there. Okay, so things have changed over the years. Okay, yeah.
But that cut and square, what it's saying is like there's a 50% chance that if it's a boy, he'll have the disease, right?
Correct.
Okay.
Yep, here's the males. So you have so half of it will be males, half will be females. Of the males, 50% will have the disease and 50% will have completely normal. And here, 100% of the females here, these two blocks here, 50% will be completely normal, 50% will be a carrier, not a disease. Right. Okay? So here's a pedigree analysis. You can do this on your own if you want to do that, but you can go back to those kind of things. But it skips a generation. Okay? So some features that sex-linked diseases have, that you put this together. Okay? Females are either normal or carriers. It is extremely rare to have a female having one of these diseases. They can only get the disease if she inherits the defective allele from both her mother and father, which is, for these diseases, it's not likely for it to happen. Okay? So it's rare for a female to inherit this disease. Males are either normal or diseased. They're never carriers. They can't be. They only got one X. Okay? And the disease skips a generation. Okay? Questions on cytology.